Okay, so we are going to talk about our very last chapter, development, heredity, and aging. So we hit on development just a little bit in terms of our progression from zygote, the fertilized egg cell, to embryo up till 8 weeks, and then fetus. We're going to focus on heredity for the most part. Here are some of the MPOs off of your syllabus that kind of relate specifically to this chapter. This chapter is a bit of an outlier just because it's not one of our body systems like a majority of our chapters have been. And we're really just trying to explain the basic principles of human genetics and heredity. So the word genetics itself is the study of heredity, and heredity is passing down of characteristics via our chromosomes, our genetic information. And so you could also consider this the study of inheritance, or how we are inheriting our genes, our traits from previous generations. And so if you remember back from when we talked about the different organelles and macronutrients or major structures that make up our body. We talked about DNA, which is one of our nucleic acids, and chromosomes. So our chromosomes are tightly wound bundles of chromatin that are housed within the nucleus. Our normal body cells, our somatic cells, they're going to contain 23 pairs of chromosomes. So you need to pay attention to this word, 23 pairs. So our sex cells, our gametes that we talked about in chapter 19, remember, only have 23 total chromosomes. And so a gene itself is what we consider a functional unit of heredity. That's something that we can study or watch as it's passed down from generation to generation. And it's a specific segment of the DNA that's going to code for the production of a specific protein. So that's why I often refer to DNA as a recipe, because it has the recipe for all the different proteins that make up everything that functions within the body. And remember, that is going to be produced, or these proteins are produced by the ribosomes. That's what reads the recipe. So if we're looking at this set here, we see we have 23 pairs of chromosomes. And so this is going to be a normal cell, a somatic cell, because you get one set from mom, one set from dad. And so a gamete would only have one pair from each of these. And so when they combine, we can get our full set, one from mom, one from dad. You'll also notice here, and we'll talk about it in just a second, that 1 through 22 are numbered, and then our 23rd pair here shows X and Y. We call those our sex chromosomes. So they're going to determine sex. In this case, we're looking at a set of male DNA because we have an X and a Y. If this was female DNA, we would have X and X. So you'll see a lot of bolded terms, this kind of a definition-based chapter with a lot of this stuff. So we have 23 pairs in a body cell of homologous chromosomes. So each chromosome in a pair is going to code for the same genes. So your number one chromosome from mom and your number one chromosome from dad are all going to contain some version of the same genes. 1 through 22, we call the autosomes. And so they're going to code for every single thing in the body other than sex. So our sex chromosomes, this 23rd pair, is going to code for genetic sex. Like I said, if it's male DNA, this is going to be an XY. For females, we have two X chromosomes for our sex chromosomes. And while they obviously code for our genetic sex, they also have other traits associated. So they don't only code for sex. There's other things involved in these chromosomes. So this is that word I was saying in the first couple slides. A somatic cell are the body, our body's normal cells in a sense. Anything besides a sperm or an ovum. And so we call those diploid because they have two pairs of 23 chromosomes. So 2 times 23 gets us our 46 total chromosomes. When our somatic cells divide, they kind of start with a full set and end with a full set. So we start with one, end with two. That process is called mitosis. Gametes are our sex cells. We saw that word in chapter 19. So, of course, for the male system, that's going to be a sperm or spermatozoa. For our female system, that's an ovum. It could be a egg cell. It could be an oocyte, depending on which term you choose. We called those haploid cells. They just have 23. We don't multiply by two because our sperm and our ovum that are both haploid will combine to make a zygote that's a diploid cell. So if we started with 46 total in each of our sperm and ovum, this zygote would have twice as much as it needs for its full recipe. So before I even jump into this aspect, I just want to point out this chart over here. Some of you may have seen this if you've had genetics in any of your previous middle school or high school classes, but we call this setup here a Punnett square. And so a Punnett square is a very simple, kind of simplified way of looking at how genetics or how genes are passed down or inherited. And what it really looks at is your probability of having a specific outcome. In this case, we're talking about sex. So we have mom on this side, an X oocyte, an X oocyte. It's always going to be an X for mom because remember, we have two XX chromosomes. So all of the eggs are going to have an X. Dad is up here on the top where the sperm cells are. And so males are XY. And so about 50% of sperm will have an X and 50% of sperm will have a Y. And because mom always donates an X, in a sense, the sperm cell is going to determine the genetic sex of the offspring. If it's an X sperm, then we're going to end up with a female. So the way a Punnett square works is you kind of follow it across, follow it down. So if we have an X in an X meeting, X in an X meeting, that's going to be two female. So 50% chance of female, XY, XY, 50% chance of male. So that's why I say dad ultimately determines the sex because mom's always going to contribute an X. Okay, some more terms to start to memorize is an allele. And so allele is kind of the term for a gene. And so it's a similar gene on homologous chromosomes. So for example, this black band and this black band on these chromosomes would be alleles. And so when we keep it really simple, we have two alleles for a particular trait. And so we can call those homozygous or heterozygous depending on the presentation. So we can have two alleles that are the same is homozygous. So homo means same. So they can either be both dominant or both recessive. So when we use the word homozygous, we have to say homozygous dominant or homozygous recessive. We'll talk about what those mean here in a second. And then for heterozygous, we don't have to put that qualifier on there because that means we have a mix. That means one allele is dominant, one allele is recessive. So what does dominant and recessive actually mean? So the dominant trait is going to be the gene or the allele that is expressed in a heterozygous genotype. So our genotype is what we have on paper. We use capital letters and lowercase letters to represent dominant and recessive. So this big A, little a, or big A would both be examples of genotypes. Now, the phenotype is a bit different. It's not what's on paper, it's what's actually expressed as that gene for a person's appearance. For example, hair color or eye color. So we said dominant is going to be expressed when we have a mixed genotype because it's the dominant trait. It's going to overrule the recessive. So big A is going to beat out little a. The other way we can show a dominant phenotype is when we have a homozygous dominant genotype. So that's big A, big A. So if we have two dominant copies of that allele, of course, we're going to express the dominant phenotype. So for example, if I said the big A stands for brown hair and the little a stands for blonde hair, in this case, on paper, you would have one allele for brown hair, one allele for blonde hair. This would be your genotype. Because the dominant beats out the recessive, the phenotype would be brown hair because that's what's going to show through and be expressed is the dominant trait. The only way we're going to see a recessive gene, our little a, blonde hair in our example, is a homozygous recessive individual. And so that would be two little a's. So there's no big A to take over. So the little a, our recessive trait, is going to be expressed. Again, in our heterozygous genotype, the big A is going to dominate and little a is going to remain hidden. We'll talk about some exceptions to that rule, but when we keep it really simple, this is how you should think about it. This table is a really good summary, so if you are feeling a little confused after that one, take a look and read through this, and that should help clarify things for you a bit. And if not, of course, please email your instructor for some extra help. All right, a couple of genetic disorders, or kind of the concept of genetic disorders, are caused by abnormalities in our genetic makeup. So that's obviously within the DNA. So this might involve an entire gene, or sorry, kind of a single gene, one small section of a chromosome, or it could be an issue with the entire chromosome, which would affect multiple traits. So, a lot of genetic disorders result from what we call a mutation. And this is a change in the DNA that usually kind of involves a change in the letter of the recipe. And so, as things were getting copied over, there was some kind of mistake in the transcription or translation. And now we have kind of the wrong recipe, which means we're going to make the wrong protein. This term, non-disjunction, this is when the homologous chromosomes fail to separate. And so in meiosis 1 and 2, that's how we get down from our 46 total chromosomes to 23 total chromosomes. So if we don't end up getting a separation, that can kind of pull one extra chromosome over with it. And so we call that aneuploidy, which means we have multiple or an abnormal number of chromosomes, more than we intended to have, or could be less than we intended to have, depending on which direction that chromosome is getting pulled. So heredity is quite complex. This example that we went through here, these terms in this summary table, really only apply when we're trying to keep it as basic and simple as possible. In reality, there's very few phenotypes where we can draw a kind of a nice little kind of four square Punnett square and figure out exactly how that's happening. Most of the time, there's multiple genes that are going to contribute to a trait or multiple alleles that are going to contribute to a trait. And so we call those polygenic traits. And that's a majority of the traits in our body. They're influenced by multiple chromosomes, multiple genes. In terms of inheritance, dominant inheritance means that these traits are carried on the dominant allele only. And so some examples that are really common that are carried on dominant alleles are widow's peak hairline, freckles, dimples, double jointed thumbs, so kind of being able to bend your thumb back towards your forearm, and the ability to roll your tongue or kind of make a circle with your tongue. Dominant disorders then are going to be the disorders that are linked with dominant alleles, but these are relatively uncommon. A lot of them are lethal and tend to result in death prior to reproductive age, so it's unable to be passed down. It's also dominant, which means it's going to show through even if it's a heterozygous genotype. And so that means that it's not going to be hidden. You can't be a carrier that we'll talk about with recessive disorders. So a lot of times, if people have a dominant disorder that is kind of going to affect your life negatively, they may choose not to pass on that or not to reproduce so that they don't risk their children having that disorder as well. Here's some examples of dominant disorders. Syndactyly, where the skin between your fingers and toes do not separate completely. Achondroplasia, that's a type of dwarfism. So this could be heterozygous or homozygous. If it's heterozygous, that's what results in dwarfism. And homozygous, dominant for this disorder, will result in death. It's not going to be viable. Huntington's disease is one that is relatively more common. And this is a really scary disease. Oftentimes doesn't present. You kind of only know that you have it because there's a genetic history in the family, and it results in really, really rapid brain degeneration. Most people, if they know they have Huntington's disease, will choose not to risk passing that on. Now, recessive inheritance is a little more tricky because our recessive traits can kind of hide in our heterozygous genotype. But there's some really important recessive traits like normal digits, normal bone formation, and not having Huntington's disease. So, dominant doesn't necessarily mean that it's the better gene, it just means it's the one that's going to show through. And so recessive disorders are much more common because, as I was saying, these heterozygous individuals are carriers. So the ones that have a big A, little a for a recessive disorder, that little a is going to hide. So the person that has that genotype may not even know that they have it because it's not going to be expressed as a phenotype. And so, oftentimes, and this is becoming more common now, genetic testing is what really reveals the presence of a recessive disorder. Or, again, if there's enough of a family history of that recessive disorder, that may kind of clue the doctor in. But oftentimes, people don't even know that they are carriers. So it makes it much easier to pass on these disorders without realizing they're carriers. They don't even know they have the disease. It's not until it shows up in their children that um, it's revealed that they're a carrier. Some examples is are albinism, which is a lack of melanin in the skin. There's Tay-Sachs disease. That's a disease that attacks our nerve cells. And then cystic fibrosis is another example of a recessive disorder that actually affects how thick our mucus is and is commonly kind of presents with breathing and lung issues. Okay, now we're going to get into a little more of the weeds here in terms of why it's not quite as simple as just dominant and recessive. We can also have something called incomplete dominance. So this is going to be a heterozygous genotype, a big A, little a, or you'll see could vary depending on which trait we're talking about in terms of what letter we use. And they're going to have kind of an in-between or an intermediate phenotype. The example we use here is sickle cell anemia. And so someone with the heterozygous genotype is not going to express or show as severe of symptoms as someone with the homozygous genotype. And so if you had two big S's for sickle gene, you're going to have normal hemoglobin. You're going to have nice circular red blood cells. They're going to fit through all of your vessels quite easily. If you're heterozygous, big S, little s, we call that sickle cell trait. So this is incomplete dominance. This big S doesn't completely take over. It's influenced by this little s. So you get a kind of a mixture or combination of mutated or sickle hemoglobin and normal hemoglobin. So they're not going to have issues all the time, but they're more prone to what we call a sickle cell crisis. And then if we go all the way to homozygous recessive, that we call sickle cell anemia, and that means you're only going to have sickle mutated hemoglobin, and they're going to be much more susceptible to sickle cell crisis compared to a heterozygous individual. So incomplete, we get a mixture or kind of an in between of the homozygous dominant and homozygous recessive. So we already talked about this idea of multiple alleles that control blood type. And so we have three different alleles that control, multiple alleles that control a single trait, but we're going to use the example of blood type here. And so we're also going to introduce this concept of codominance. And so that means that both phenotypes are expressed in a heterozygous individual. So in blood type, we have two dominant alleles, I with a little a, or sorry, big A, I should really say, and I with a big B. And then we have little I. So this is going to be people with A blood type, B blood type. And if you have both, that's how you become type AB. So it's not that one is preferential over the other, they both show through. If you have little I, so type O blood, that means you end up with two little I alleles for blood type. So you're not necessarily going to be tested on figuring out blood type in terms of this Punnett square here, but this kind of shows how this codominance works. So if we have IA with little I, that's type A blood because A is going to dominate over I. If you have IB with little I, you're going to have type B blood because B is going to beat out little I. The only way you can have type O is two little I alleles. And then for homozygous dominant, if we have AB, that's where the codominance comes in. So that's how you get type AB blood. And then, of course, if you have two IAs, type A, two IBs, type B. In terms of this Punnett square, it works the same way in terms of kind of following the rows and columns. So it's just adding some more boxes, but it doesn't necessarily complicate things. Okay, one of the last things that we're going to discuss is sex-linked traits. And so our X chromosome, like we said, both the X and the Y carry more than just our genetic sex, but the X chromosome especially has a lot of genes. And so kind of makes sense that we all have at least one X chromosome. Our Y chromosomes are important, but they're quite tiny. They only code for about 200 genes. And so we have some genes that are only found on our X chromosome, and we call those X-linked genes, which are typically the sex-linked traits we are discussing. So, because females have two X's, oftentimes a X-linked gene, unless it shows through on both X's, is not going to be expressed in a female. In our male combinations, we only have one X. So if that gene is only found on the X chromosome, then it is going to be expressed in males. Kind of common example of this is color blindness, and this is one that you'll see in your genetics lab, where there's really no colorblind females, or it's quite rare to have colorblind females because it's an X-linked trait. So even if one of the X chromosomes has the colorblind allele, they have a second X that can kind of dominate and overrule. In the males, because they only have one X, if they end up with that recessive colorblind trait, their Y chromosome does not have an allele to overrule that. So they're going to show through with color blindness. So this is kind of what I was just talking about. So X-linked recessive alleles are always expressed in males because there's only one X. A female would have to be homozygous recessive on the X chromosome to express an X-linked condition. So X-linked conditions tend to be passed from mothers down to their sons because the mother is heterozygous. She's a carrier and she doesn't even know she has an issue. Hemophilia is excessive bleeding, so that would be one that you would definitely not want to pass down if possible. And then color blindness, not as big of a deal, of course. So it can be passed from mothers to daughters, but like we said, you'd have to be homozygous recessive for that to be possible. All right, everyone, that is the end of chapter 20 really focus just on the depth, kind of getting those definitions down and understanding your different types of genotypes and what that means about phenotype. Thank you guys for a great semester and try to finish strong on the last couple exams.